Welcome to this video on cell division, specifically mitosis. We'll cover the cell cycle, chromosomes, DNA replication, and the stages of division, essential knowledge for your biology studies. Here is an overview of what we'll cover today. We will start with why cells divide, then look at DNA and chromosomes, followed by the cell cycle and the stages of mitosis. Finally, we'll discuss uncontrolled division and some exam tips. Cells divide for three main reasons: growth, repair, and asexual reproduction. Growth involves producing more cells to increase the size of an organism. Repair replaces damaged or worn-out cells. Asexual reproduction produces genetically identical offspring. The key rule of mitosis is that it always produces two daughter cells that are genetically identical to each other and to the parent cell. This is vital because replacement cells must carry the exact same genetic instructions to function correctly. Before diving into mitosis, it is important to understand where genetic information is stored. This brings us to DNA and chromosomes. DNA carries the genetic instructions for an organism. It's found in the nucleus, tightly coiled into chromosomes. Human body cells have 46 chromosomes, arranged in 23 pairs. The nucleus is where these chromosomes are located. The cell cycle is a regulated sequence of events leading up to cell division. It consists of three main parts: interphase, mitosis, and cytokinesis. This cycle ensures division is orderly and accurate. Stage 1 of the cell cycle is interphase, the longest phase. During this time, the cell prepares for division through a period of intense activity. During interphase, the cell grows larger, producing more cytoplasm, energy, and raw materials. It also duplicates subcellular structures like mitochondria and ribosomes. Crucially, the DNA replicates, ensuring a complete set of genetic instructions for both daughter cells. DNA replication is critical because both new cells must receive a complete, identical set of genetic instructions. Each original chromosome pairs up with an identical partner called a sister chromatid. Remember, DNA replication happens during interphase, before mitosis begins. Once interphase and DNA replication are complete, the cell enters stage 2, mitosis. This is the process of nuclear division, which is further divided into a series of distinct stages. The four main stages of mitosis are prophase, metaphase, anaphase, and telophase. The entire process is continuous, but dividing it into these stages helps us study and describe the complete division of the cell's nucleus. Stage 1 of mitosis is prophase. Here, duplicated chromosomes condense and coil up, becoming visible as distinct X-shaped structures. The nuclear envelope begins to break down, allowing the cell's machinery to access the chromosomes. Stage 2 is metaphase. Chromosomes move to the center of the cell and line up along the equator. Spindle fibers attach to the centromere of each chromosome. This alignment ensures each new cell receives one copy of each chromosome. Stage 3 is anaphase, the big separation. Spindle fibers shorten and contract, pulling the sister chromatids apart towards opposite poles of the cell. The separating chromosomes often form V or Y shapes, a classic image in exam diagrams. Stage 4, telophase, marks the end of nuclear division. A full set of chromosomes arrives at each pole, uncoils, and becomes less distinct. A new nuclear envelope forms around each set, resulting in two separate nuclei within one cell. Mitosis divides the nucleus. Cytokinesis is the final stage, dividing the rest of the cell, including the cytoplasm and cell membrane, to produce two physically separate daughter cells. In animal cells, the cell membrane pinches inward to form a cleavage furrow. In plant cells, a new cell plate forms across the middle, eventually becoming a new cell wall. Cytokinesis completes the M phase of the cell cycle. Mitosis produces two daughter cells, each with 46 chromosomes, which are 100% genetically identical to the parent cell. This genetic fidelity is crucial for growth and tissue repair. Every cell in your body, from your brain to your fingernails, contains the same DNA. This identical DNA is a powerful concept resulting from continuous, accurate rounds of mitosis from the original fertilized egg cell. Here is a quick overview of the entire cell cycle, including interphase and the stages of mitosis: prophase, metaphase, anaphase, and telophase and cytokinesis. Use this summary to check your understanding of the whole sequence. Mitosis is also the basis of asexual reproduction in many organisms. When a single parent produces offspring using only mitosis, the offspring are genetically identical to the parent, often called clones. Examples of asexual reproduction via mitosis include yeast budding, where a new, identical cell pinches off. Starfish regeneration, where a new body grows from an arm, and strawberry runners, which form new plants. Asexual reproduction is fast, energy efficient, and transmits all successful genes. However, a disadvantage is the lack of genetic variation, which makes the entire population vulnerable to disease or environmental change. The cell cycle is tightly controlled, but if this control breaks down, the result can be cancer. Cancer is fundamentally about uncontrolled cell division. Cancer occurs when cells divide uncontrollably, often due to mutations affecting cell cycle regulation. This forms a tumor. Mutations can be caused by carcinogens, replication errors, viruses, or inherited faulty genes. Normal cell division is carefully controlled, dividing only when needed, and undergoing apoptosis when faulty. Cancerous cells ignore stop signals and divide continuously, forming tumors. Understanding this control system helps develop cancer treatments. To summarize, the cell cycle is a continuous, repeating process of growth and division. Each full cycle, which includes interphase, mitosis, and cytokinesis, produces two genetically identical daughter cells. Let's quickly review some key vocabulary. Mitosis is cell division producing two identical daughter cells. A chromosome is the thread-like structure of DNA in the nucleus. DNA replication is the copying of DNA before division, and cytokinesis is the division of the cytoplasm. Interphase is the growth and DNA replication stage before division. Sister chromatids are the identical chromosome copies separated during anaphase. A tumor is a mass of uncontrollably dividing cells, and asexual reproduction involves a single parent producing identical offspring. Now that we've covered the science, let's focus on how to use this knowledge to ace your exams. Knowing examiner expectations gives you a real advantage. First, always state that mitosis produces identical cells. Remember that DNA replicates before mitosis, during interphase, not during division itself. Also, link cancer to uncontrolled cell division. Avoid common errors like confusing mitosis with meiosis, or describing cancer simply as fast division. Instead, examiners want to see key terminology and the correct sequence of the cell cycle: interphase, mitosis, and cytokinesis. Mitosis produces two identical cells for growth and repair with the same chromosome number. Meiosis produces 4 varied sex cells with half the chromosome number. Remember this key distinction to secure marks. To recap, mitosis creates identical cells, DNA replicates first, the cell cycle follows a fixed order, and cancer is uncontrolled division. Mastering these points will ensure your success in this topic.